Hello. Today I will be briefly discussing the general workflow required to utilize bioinformatics into the genetic variant characterization process. I will start off by briefly listing the methods used to elucidate an answer once you have a desired protein or gene of interest. The initial step is to build the protein structure inside of a molecular modeling simulation software known as Yosara. Once you have your initial template structure, you will build out any missing areas using a different program known as iTaser. Once we have a reliable model, we can start to pull known variants off of either clinical databases, or if we have a patient, we can take their specific variant and model it onto the model. And here are just a few examples of several of the models that me and the research team have modeled up to this point, with a heat map of the most common variants found in human disease up to date. So the current focus of my research, as well as my mentor's research, is on a list known as the ACMG 59 list. This list is a list of 59 genes that has been published by the American College of Medical Genetics that all show high penetrance and actionability. Basically, these are the go-to if you want to make a difference in the world of genetics right now. What we do with these 59 genes is a combination of protein modeling, molecular dynamics, and pathogenicity prediction tools to understand basically the cause of disease, going off of the genetic mutation that's causing it. A secondary goal of this research is to clean up a website known as ClinVar, which takes all known clinical variants and assigns a pathogenicity prediction score to them. And what we have discovered is that a large majority of them are misclassified at this date. And I know this is hard to see, but here's just a breakdown of the diseases associated with the aforementioned 59 genes on the ACMG 59 list. Here's a more zoomed in view of a structure. This is actually a mutation that was found in a patient during my time at Michigan State University's medical school this past summer. And here is a figure that kind of correlates to that previous statement I made about ClinVar misclassifying variants. As of right now, you can see about a quarter of the variants in ClinVar are determined to be disease-causing or pathogenic. Roughly 3% have been defined as benign, which means they have no influence on disease. The other 3/4 of variants out there right now, no one knows what they mean or what they do. My goal is to get that 75% much lower so that we're not just providing variants of uncertain significance, but can actually start to answer the questions regarding whether a mutation is causing the patient's disease or not. So what is the plan now that we have all these structures and all this ClinVar data? We want to determine whether the variants that are classified as pathogenic are actually disease-causing, or if that is a false statement. The way we do this is by running molecular dynamics simulations on these genes, which basically looks at the movement of the wild type or non-mutated version as opposed to the mutated version, and see if there's any difference. We then can provide a guide that shows the best tool to utilize for these specific genes, and what I mean by that is what softwares to use to get to this answer and which ones to steer away from. And very briefly, I'm going to show the next steps beyond this once we've maxed out our computational resources basically and advancing it into the wet lab. Another thing I did over the summer was developing an induced pluripotent stem cell to beating cardiomyocyte differentiation protocol. And here's just a few pretty pictures of those basically. And here's actually a video of them beating on the Petri dish. All right. And that is all I have for now. If you have any questions, feel free to contact me.